Great, thanks a lot. I'm going to be talking about some...
I'm sorry. After this, we are going to have a break, and then we are going to have a short break. And after that, we are going to have a session where we are going to be showcasing 3 of our 8 postdocs, Simon's Fellows for the Evolutionary Biology program.
All right, so I'm going to be talking about some aspects of bacterial growth in fluctuating environments coming from experiments we've been doing in the last couple of years. And I'll try to highlight also some theoretical sides of this, but most of the talk will basically focus on experimental results, and we'll sort of go from systems biology of responses of bacteria and eventually towards small populations. So basically, we've been measuring E. coli population dynamics at the single cell resolution, where we can track all the individuals in a population while controlling the environment. So that's the kind of experiments we set out to do. And today I'm going to talk about 3 aspects of that we've been studying. One is the responses that you see when you expose cells to fluctuating sugars. Then I'm going to talk about the statistics of the lineages that you see in small populations in constant environments. And at the end, I'm going to talk about what this looks like when you have small populations that are experiencing fluctuating stresses. So the device we're using was built by a postdoc in the lab, Guillaume Lambert, and most of the work I'm going to talk about today was done by Guillaume. And this is really a modification of something that's called the mother machine. And that was invented in 2010 by Seok-Jun Jun. It's a microfluidic device in which you can grow single cells of E. coli in small lines such that the mother cell remains trapped and you just continue to observe cell divisions for long periods of time. And so we thought this would be a great device for doing fluctuating environment experiments with single cell tracking if we could expand those chambers and make them slightly larger so that we have small populations. So that in principle, the effective population size here is larger than 1 cell, but it's certainly still a small population. And so the device works in the following way. You have a flow of nutrients that brings in, which is bringing in nutrients and also taking away cells. And the nutrients get into these chambers purely by diffusion. And the nutrients we're bringing in are small sugars, so the diffusion time and the turnover time of the chemical composition of these chambers is on the order of a few seconds. And we can actually measure this by flowing in small fluorescent molecules. So we can basically feed all these cells happily and watch the cell divisions here. So I'm going to show you just a couple of examples of what we see. So the chamber sizes, you can see sort of from the size of E. coli, which here are roughly 1 to 2 microns in size. This is minimal medium. They're growing on pure glucose, and so the cell size is roughly between 1 and 2 microns. If you grow them in rich conditions, the cells get larger. And so in this experiment, we're following 5 separate populations in constant glucose, and then we're just going to fluctuate after 4 hours to no glucose at all. So here you're watching cell divisions for 4 hours. Now you fluctuate to no glucose, and the cells stop dividing. And then once you supply glucose again, they continue to divide. And you can keep these cells in focus for a long period of time and track all the cells and continue this kind of experiment. We can also do other things like stressing the cells by giving them pulses of antibiotics. So in this case, we're giving pulses of carbonicillin, short pulses, and every time you have a pulse, you kill cells. These are 20-minute pulses, and so the population can still continue dividing because the rate of growth in the times in which there's no antibiotic is sufficient to maintain a population size, and I'm going to talk about this dynamics a little bit later. So the first thing we started looking at is what happens when you change the carbon source that the cells are growing on. And so this really goes back to some pioneering experiments at the beginning of gene regulation, which were done by Monod in bacteria. And Monod's observations are well known. If you shift bacteria from one carbon source to another, typically the cells have to go through a lag, and eventually this led to the discovery of gene regulation. So basically in this experiment, he was growing cells on pure arabinose. If he switches them to glucose, it turns out they don't go through a lag. So they already have the machinery to digest glucose, and so they grow exponentially in glucose essentially immediately. If you switch from one sugar source to another, like if you switch to xylose, then you go through some lag, and eventually the cells, after, in this case, 2 to 3 hours, they are able to recover full growth, exponential growth. Also, this is another well-known observation. If you grow them on mixtures of 2 carbons, they preferentially digest the glucose. So the more glucose you give the cells, they rise to a higher density before plateauing. And the length of this plateau depends very much on which sugars are involved, but typical time scales here are 1 to 2 hours, depending on the carbon source. So we can repeat this in our device, and so now if we grow cells in glucose, you see them growing, and we're going to fluctuate to lactose. Right there, the cells stop dividing for about an hour, and then they can resume growth. So that's a single fluctuation from glucose to lactose. And what goes on actually in the cells here is that the cells are turning on the genes to metabolize lactose. So we can, in this case, quantify things. Even without tracking single cells, we can simply look how fast are cells pushing past a specific cross-section of this device, and use that as a proxy for the elongation rate of the population. And so if you plot this cell speed of just pure pushing, keep in mind this is just cells elongating and pushing against each other, you're going to see in glucose that you're in some pushing state here, some equilibrium cell speed, and then when you fluctuate to lactose, you basically go directly to 0. There's no cell growth at all. And after about 30 minutes, you can now slowly resume growth, and it takes about 50 minutes to get back to normal. And now the cells can grow happily in lactose. So that's a single fluctuation. And what we did is we continued this. So we always see this lag in recovery. We continue this now in fluctuating environments. So we do 4 hours of lactose followed by 4 hours of glucose, and we continue to fluctuate. And what you see in this case is that the lag, which you initially encountered, disappears. So the next time you encounter lactose, you never go through lags, and the cells can now comfortably grow on both glucose and lactose in this fluctuating environment. So the time scale here is 4 hours of glucose, 4 hours of lactose, and in this condition, the cells are growing with a rate of cell division, which is an hour, which means that in 4 hours, the cells have gone 4 cell divisions. So if you think of this as a small population, you have basically 4 generations between these fluctuating environments. So what this suggests is that the cells can remember the past through 4 cell divisions.
They're growing essentially as fast as they were just in lactate, just in glucose?
Yeah. Yeah, that was also surprising. So now we can play with these time scales of how long you grow between exposures and look at this memory. So we vary the time since the lactose exposure, and I showed you that at 4 hours you have no growth delay in the second exposure, so that's perfect memory. We call that phenotypic memory. There's no lag or recovery phase in this case, and that's true if you fluctuate certainly faster. Now if you go longer than 4 hours, then you start forgetting, the cells start forgetting that they've already seen lactose, and clearly if you grow them for very long periods of time in glucose, they completely forget that they see lactose, and so they have to go through a complete lag again. And so that's this kind of
the same size as the original size.
Eventually, it will be of the same size if you let them grow for long, long periods of time.
Sorry, what's the difference between the lag phase and the recovery phase?
The lag phase is the dead time where you absolutely do not grow, and there's a slow, right? So this is how these curves, basically this is the forgetting, right? This is the complete forgetting of memory. So if you estimate, well, I mean, this is really the curve, but you could say that, you're going to see a significant difference between, cells which have never seen lactose because we've grown them for many, many generations without lactose, and 12 hours of growth. There is still some difference here. So roughly, it seems that some part of this memory can persist for up to 10 generations. And here we see that you get really perfect memory for 4 generations. So what is the basis of this memory? And yeah.
Are those all a single shift or actually repeated shifts?
Yeah, so that's this kind of experiment, basically. So what I was just plotting was the time to recover after the second glucose exposure. So we went through the molecular side of this. I was going to just briefly mention it. I can certainly say more about the molecular details, but it's good to just get a sense for what can provide this kind of memory in the cells. The key genetic network here is the lac operon, which turns on the genes to metabolize lactose. And that network has been studied in immense detail. The key gene for metabolizing lactose is called LAC-Z. It's beta-galactosidase. The gene for bringing in, the protein that brings in lactose is called the lac permease, LAC-Y. These are produced off of a single messenger RNA. And these genes are repressed by a protein that's called LAC-I, which is the lac repressor. That protein, when it senses lactose, in particular it senses allolactose, but that's a side story, it falls off of DNA and it allows production of this operon, production of these genes. There's a positive feedback loop here because permease is now produced, brings in more lactose, which means that the probability of unbinding of lac repressor increases, and you have this powerful positive feedback loop which then rapidly turns this gene on. So what provides memory in this system? We find that in fact the lac proteins themselves are the memory. So the cells maintain the proteins. They, in particular LAC-Z and LAC-Y, each provide memory. They provide different parts of this memory. LAC-Z, in fact, on its own can completely eliminate lags. We see that here. And LAC-Y, if you already provide it, it shortens these lags. And we can also follow in single cells by labeling, fluorescently labeling the permease. We can actually see the dynamics of the permease in the cells. And we can actually see that if you have enough time, of course, you dilute this permease, okay, and then you have to go through a full lag. But if you make faster fluctuations, you never fully dilute this, and that apparently is sufficient to maintain growth. So the proteins themselves are super stable proteins. Their degradation times on their own are in the 1 week time scale, which means that the only reason that you see dilution here is not because of active degradation of the proteins, but just because of cell growth and elongation. Yeah?
Why does the cell turn this thing off anyway? I mean, even if it doesn't get the other kind of sugar, it could keep it on running. What's the cost of it having to turn it off if it just gets glucose?
Yeah, so those costs have been measured, and it depends sensitively on the conditions that you're in. But people have measured costs for having this operon on. It depends on how much of it it's on, and it really has to do with the fact that to bring in lactose, the lac permease actually uses the proton gradient, okay, across the cell membrane. So for destroying that gradient is not good for the cell in general. So, okay. So this phenotypic memory, as you can see, is useful for fluctuations which are on intermediate time scales, 1 to 10 generations we're estimating here. Now we also found this memory, I'm not going to show these results, but just tell you that we found this memory also in other sugar operons. So for lactose, galactose, arabinose, maltose, in all these cases you have this kind of phenotypic memory. So next we asked, okay, so that gives you intermediate scale. What about if you now fluctuate faster? So imagine that before you finished adapting to lactose, normally you go through this 30 minutes of dead time and then this recovery phase. Imagine at this point somewhere, you now fluctuated back to glucose and you do faster fluctuations. Or even imagine something more extreme. Before you even left this lag phase, you now switch back to glucose and see what happens. So in that case, we actually find a different kind of behavior. I'll mention this in a moment. So what we see is when you fluctuate from glucose to lactose, you go through this 30-minute dead time. Now you go back to glucose, you can already grow in glucose, okay? And now when you go back to lactose, you can grow perfectly well. So keep in mind, you haven't seen lactose here for 30 minutes, but as far as the lac operon, you've somehow gone from 0 to induction through the glucose exposure. If you go faster and you see lactose for 10 minutes, then glucose, by the time you see lactose next, you're already in the recovery phase. You see glucose for another 10 minutes, and by the time you got lactose again, you're perfectly growing in lactose, and you've only seen lactose for a total of 20 minutes. Okay, so if you go through glucose exposures, you actually adapt. Turns out you adapt faster, which means that the cells are remembering the process of responding. So that's what we call this a response memory. Okay, so this result, we can also look in at the molecular side of it, and I'm going to go quickly through this, but basically we can give pulses of lactose and look at how fast the cells turn on the genes for the lac operon, and we see that after you remove lactose, there's an overshoot, which peaks after 40 minutes. So the cells continue to respond to lactose even after you've removed it from the medium for about 40 minutes, and that gives you this kind of response memory. And we can go further, because in the lac operon's been studied very, very well. There's a lot of tools to play around with it, and we can show this really boils down to the lac repressor determining this response memory profile. So you can actually shift this response memory profile by changing the affinity of the lac repressor for DNA, so you can actually make it turn off faster. Okay, so it seems that certainly the properties of the binding of the lac repressor can in principle be tuned to give you different response memory profiles. Okay, so on this memory side, we see that in the fluctuating environments, you have two types of memory that can be useful. One is this phenotypic memory, which is this passive passing down of proteins, and that's useful on these intermediate time scales. And then you can have response memory, which would be useful for faster fluctuations or pulses. So now to say something about the implications of this, if you think about what determines gene expression levels in evolution, how they evolve, typically people think about cost benefits. So the cost of producing the protein versus the benefit of having it when you need it. Here, this adds another layer to it, because it could very well be that you're actually overproducing the protein to some extent in any given generation, so that the progeny will avoid lag phases. And so incorporating that into some analysis would also be interesting. Is what are the implications of having memory for the evolution of gene expression profiles? Okay, so that was the first section on responses to fluctuating sugars. So now I want to go to the next step, which is that instead of just looking at growth rates, we actually have a lot more data in these devices. We have all the single cells, and they're being tracked. So it'd be interesting to see what do the statistics look when you look in finer detail now at the kind of lineages that you get.
Yeah. You mentioned that it could be that it is producing extra proteins for the progenies, or it could be just the time lag. It could be just a time lag to get the protein out, so there could be an inertial effect, right? It doesn't have to be with any cause. Is that what viable, you think?
Yeah, I mean, it's really a question of, what determines the gene expression levels? How are they set? How do they evolve? And so what I'm saying here is that there's another layer to it, which is that you may not just be adapting to the here and now, okay, but also your expression levels could be set by the fact that they allow your progeny to avoid going through longer lags. Yeah.
So in terms of learning, what you've shown is that they're capable of sensitization, and you've got a very good system in which you could try and investigate whether they're actually capable of associative learning, to see whether bacteria can be classically conditioned.
Yes.
As far as I know, the simplest organism that can be classically conditioned is a paramecium, and that's a debatable result.
Right.
But you could definitely easily try it out. I've got a couple of papers which show how they could be classically conditioned, so I'd really be interested to see if you could demonstrate
Absolutely.
that.
Great. Okay, so now to go into this lineage, so I want to talk a little bit about the branching process of cell divisions. These are results. I'm going to discuss for the next few slides results from a slightly different device in which we maintain even smaller populations of 10 to 20 cells, and we track them. And then I'll go back to our larger device in a bit. So here we really have single cell tracking of these small populations of 10 to 20 cells. We're growing them in constant conditions in minimal medium, and we're observing them for 3.5 days, so that's about 100 generations of cell divisions. And this is the tree from one such population. And so let me just say what I mean by this tree. If I zoom in here on this piece of data, then this is the tree. And this shows you when the cells divided. So the branch length here is the cell division time. And every time a cell leaves this device, then the branch ends. Okay, so this is not death, this is just flowing out of the device.
But you then kill it afterwards.
Yeah, it will eventually die.
But doesn't the geometry then bias this tree, the geometry of the device?
It certainly could, right? So in spatial, trees in space will have different shapes.
Right.
That's true. Okay, so now we have this big tree, and the first thing we can do is ignore the tree and just ask about the branch lengths, which is really the cell cycle. So this is the cell cycle distribution of E. coli in this particular condition, and I call that G of tau. And this is the time between cell divisions observed in every single cell in this population, which is about 1,700 cell divisions. So so we have this distribution, and now we can ask, so what about divisions if I look now into the tree? Okay, so I take a single cell lineage, and what I mean by that is I look at the cell and now I just track it backwards. And so it goes through a number of cell divisions back to its ancestor. Different cells will have different trajectories, and of course over time, right, we're gonna, by coalescence, we're coalescing on to some single trajectory here which remains after long times. So the question is, what are the...
Is my guess correct that the spine of this is the inner chamber of your...
So actually in this case, our device has, the device I'm using here has cells leaving from both sides. Okay, so it looks like this.
I see.
So the cells can go out either way. And so that's right. So the cells that remain for a long time, well, it depends on where they moved around. Right. Okay, so now I'd like to know, what are the statistics of cell divisions along this kind of lineage? Are they different, or are they the same exact statistics as you observe in this G of tau in the tree? So a lineage in isolation would be what you get from the mother machine, which is basically trapping a single cell and just observing divisions for a long, long period of time. And in that case, every single cell comes from this G of tau. I should tell you that there is essentially no memory of these taus between generations. So you can really, to seemingly very good approximation, think of each one of these taus as being drawn independently from the same G of tau. Okay? People have measured that as well. So, okay, so those would be a lineage in isolation. Now you're in a population, okay, and the question is, do the lineages in a population look different? And so, first thought you'd say, well, they're just independent, right? So maybe not. But in fact, you should think that every time the cell divides, it makes a copy of the lineage. And so it turns out that if you consider this, you will see that in fact there's optimal lineages which will outcompete all others. And let me just say what I mean by that a little more precisely. You have this distribution G of tau, and so over long periods of time, the number of lineages which will have G of tau, okay, as their statistics, right? Because the lineages could have these intervals arranged in any possible order, the number of such lineages is going to grow as the entropy of this G of tau. At the same time, if I deviate in my statistics a little bit from this G of tau, I'm going to have slightly fewer lineages, which I can compute by how much my special G of tau is moved, right? But I might have slightly more offspring. Every single lineage might be represented with a higher number in the population, and that can also be computed. And so it turns out that what's going on here is actually a trade-off between exponential amplification, right, and the statistics of how many lineages actually are there. And so you can write down this optimum, and so this takes the flavor of a path integral, and you can compute what are these optimal lineages. And so their statistics, in fact, are G of tau modulated by an exponential that kills the tail of this G of tau at the rate which is the growth rate of that population. Okay, so we described this theory in this paper here. And then recently we actually measured this in our device, and we looked at these lineages that we get, and we measured the cell cycle age distribution along the lineages. That's the gray curve here. That's data. And then we take the prediction from single cell in isolation. You see that this red curve, it does not fit the data particularly well, and it certainly doesn't fit the tail. And then our prediction for optimal lineages would be this. So keep in mind this is a prediction, it's not a fit. It's simply that g of tau modulated by this exponential. So we can also see the dynamics of how long it takes to reach this. And indeed, you have to wait a good number of generations to get to this equilibrium.
This is not a feature of bacteria. This is a feature of sampling from the distribution of phylogeny.
Yes,
Yes,
that's
that-
right.
But it checks your independence assumption,
That's correct.
because this would look quite different with
That's
all
correct.
the subtle correlations.
Absolutely.
Just a short question about what you said about the independence. I'm a bit surprised. Like, isn't it that E. coli, when in its exponential growth, are kind of like producing, replicating their genome for a few divisions forward? You might expect like an anti-correlation between generations on the short term or something like that.
That's there is that effect, right? Which is that in LB, in rich medium, the cell division cycle is such that you're already replicating the DNA of the daughter and the granddaughter in the same cell. And so in that case, I don't remember if there are correlations. This is minimal medium, so that's not happening in these cells. Okay?
There can also be anti-correlations between the two offspring, but that doesn't enter it.
That doesn't enter. That's right.
If the cells could somehow figure out how to stay in the middle of those tubes, they would have a tremendous selective advantage, right?
They would have a purely spatial advantage. That's correct.
So I wonder if you might somehow be selecting for bacteria that figure out how to stay in the middle of those tubes.
That could happen if they could become sticky somehow in our device, but it turns out they're not. In fact, they've been knocked out. All the genes for being sticky have been knocked out in this strain. So we're trying to prevent them from doing that. There's 3 genes that make them quite sticky. Okay, so that's lineages in constant environments. And that's basically the statistics you expect in the absence of selection. So if you don't have, this is really just statistics of branching. So now we can look also at small populations under fluctuating stress. And what I mean in this case is going to be antibiotics. You saw we could give them these pulses. And let's take a look at, now we go back to our bigger device because we want to be able to somehow maintain the cells even despite the fact that we're killing them. So we need slightly bigger devices. And so this is okay, I think the background is a little blurry here, but in the back here are trajectories of 20 populations, and this red curve is the average. And so these populations that are being tracked are growing to roughly a size of about 50 or 60 cells here, and then we give them these little pulses of carbonicillin for 20 minutes, and then you see that we kill them, and then they can recover back in LB, which is rich medium, and then we kill them, etc., until it turns out they reach some other stable equilibrium here. Of course, eventually they'll go extinct, but this is some metastable state that persists. So now we look at a few things in this population. In this case, we track all the cells, and so we get this kind of picture. And we have our single cell statistics. We can look at statistics in single time slices, or we can go backwards and look at statistics on lineages. So what we find is quite interesting. If you look at this experiment and plot, again, ignore the tree and just plot the single cell statistics. So for example, the age of division, which is the g of tau I showed you before, you get this brown curve here. You see it has 2 peaks, okay? The first peak is at around just below 20 minutes. And then there's another peak here. And what it turns out, if you look in detail in these movies, is you see that cells can actually grow extra large, and then they can make 2 divisions in rapid succession. Okay, so that's what you get here, 2 divisions in rapid succession, giving rise to cell division ages which are much faster than you'd expect, right, for E. coli, which is supposed to divide it every 20 minutes. So that's one feature of this distribution. If you look at elongation rate, which is just the rate at which the cells are elongating in the population, then you see this kind of distribution here. Okay. Now if you look in the single lineages, the lineages that persist for long periods of time in this device, you get a different distribution of this age of division. And you see that it eliminates this tail. It eliminates this peak here. And if you look at the elongation rate, you see that it's in fact shifted to slower elongation rate. So what we're seeing here is that in the fluctuating condition, which involves pulses of antibiotics, the lineages which succeed are the ones that were growing slightly slower, which is the opposite of what you'd expect, right? And so the reason is, so now we have to think about what that means. So it seems there's an advantage for slower growth. I think it was mentioned in one of the first talks, is it always good to divide quickly? In bacteria, it turns out that under different types of stresses, it's actually good to divide slowly. So the very well-known nice paper on this was from Natalie Balaban in 2004 that showed that there's this phenomenon of persister in E. coli, which are cells that are basically not dividing. They're dividing very, very slowly. And if you look at these movies, you see most cells are growing fast except for these slow guys. And then you can kill everyone with an antibiotic. And the only cells that survive this are the persisters, the guys that were slow growing to begin. And since then, it's been shown in many other cases that slow growth is protective. Okay? And there's different examples of this. There's an example in yeast from Sasha Levy and Mark Siegel. There's other examples of resistance to phage. And there's a very nice review of all this from Natalie and several other people who have been working on persistence. So there's many cases and a lot going on in that. And so what the data I was just showing you suggests was that the lineage statistics suggest that in this case, instead of just two states, okay, persister or non-persister, there could be a continuum of growth rates that this kind of fluctuating selection is acting on. So the last point I want to make is that there's also a counter possibility, which is that the cells are directly responding to this changing environment, just like I showed in the cases of responsive to sugars. So the remaining question would then be, is the reason these slow-growing cells are seen in this experiment, is it because of selection on the diversity of growth rates, or is it because the single cells are making a switch to slower growth responsively, already responding to this? So the final question is really, can we distinguish these responses from selection? And so to address that, we've developed some theory previously on this. How do you distinguish responses from selection in these kinds of experiments? So the thought experiment we came up with was, imagine you can change the historical conditions of your experiment a little bit so that all the growth rates are increased by a small percentage. So in other words, you take all the FIs that go into your model or go into the actual physiology, and you imagine you could do the experiments where you give everyone a 1% bonus of fitness. And then you just rerun your experiment, right? And you see, has there been a major change of the population structure or not? And so this would be a way to detect the action of selection, because you've simply perturbed the selective strengths, right? And you're seeing whether there's any change. So that's a thought experiment. Now, to do that, you need to be able to manipulate all these growth rates in an experiment that's not going to be robust. But it turns out, at least in models, you can show a nice relationship between the average fitness of lineages that you would have upon the change in the, so this F here is the fitness along lineages, which is just the integral of the growth rates of cells along the lineage, and then averaged over the population. Now you make a perturbation of the growth rates, and you can show that the change in that average fitness of lineages will be given by the standing variance of fitness in the population in that experiment. So further details on the theory side are here, but the general idea here is that the lineage fitness variance is going to allow us to detect in these kind of experiments, we expect the strength, the effective strength of selection as balanced against physiology and response. So just to give you a little intuitive feeling for that, imagine you have a two-phenotype switching model, just two different cellular states. One is good in the first environment, the green, and the reds are good in the next environment, and they proliferate in this environment. So if you simulate this under two different models where you either have responses or you have just stochastic switching, then you'll get different kind of histories. And you can see that these histories, in which case if you have a response, they really, of course, nicely match the environment, right? Every time, these are single cell histories now from a simulation. Every time you see a tick here, the cell divides in the lineage, and the internal state of the cell is shown in the solid curve, and the background is showing you the environment. So you see if the cells match the environment, they're doing well. And these guys are responding, so they're switching. Every time the environment switches, these guys are switching. And so if you calculate, of course, the fitness of these lineages, you're going to get some value, and you see that it's quite tight because the cells are doing very similar things. If you have a stochastic switch where you just randomly can switch with some rate to the different states, you're going to see something quite different. In fact, you will see some lineages that do remarkably well, right? They almost look like they're responding, but in fact, this is just because of selection acting in the population. But at the same time, you can't do this perfectly well all the time, so parts of the lineage will also look like they're making mistakes. So you'll go through the wrong state for some time. Okay? And so the statistics here, the statistics of fitness along these lineages, are going to have a much bigger variance. Okay, so that's what we can use to detect, in principle, to measure the effect of selection. Small variance versus large variance. Now in models we can of course do everything and calculate things analytically, and things look nice. In experiments, we basically need some kind of control. So we said, can we find a control where we have, right, we would like to measure this distribution of linear fitness, okay, for example, the number of cell divisions in a lineage across a population of fluctuating environments. We need to compare this with something that we know is responding. That was our idea. Let's start with something that we know is responding as a reference. So we actually found that you can actually see by eye, I'll show you in a moment, that cells filament responsively under low doses of antibiotic. All right, so if we reduce the dose of antibiotic, the cells will actually filament, and you'll see that nicely here. You're going to get carbonicillin pulses, and now as you go along, every time you see this word carb here, look at the cells and you see that they all elongate. Okay, and you have to stare at this for a while. Okay, and you see that every time you get this carb, they just all start elongating a little bit. Okay, and then they do crazy things as well. So that's another story, right? They do all these crazy things. So this was our idea, was to say, okay, let's look at the lineage fitness for low dose versus high dose. And in low dose, we believe there's a response here. And so what we find is that the division rate along lineages in these populations, comparing no antibiotics, which is rich medium, with low dose carb, in which we think there's a response, we see they have very similar distributions. Their variance is very similar. The only difference is the mean division rate goes down, which makes sense, of course, because in this case, they're experiencing antibiotics. So here we basically have what seems to be a response. The major difference is just the shift of the mean of the lineage division rate distribution. On the other hand, if we look at this high pulses of carbonicillin, and they're still delivered at the same time window of 20 minutes, and we do the same thing, we look at the distribution of cell division rates along lineages, we see a wider variance, and we see this tail of low division rates here. Okay, so here it seems that the adaptation to this high-dose fluctuating environment involves additionally some selection on the whole continuum of cellular growth rates. Okay, and so to end, I want to show you one. Yeah.
Why you say it's selection? It is, from what I understood you just said before, when you get these long things, you get a few rapid divisions. So it's not selection in the sense of evolution and fitness. It's just you're messing with the population and you change your distribution. That's selection.
Right. That's okay. So that would be possible. In our case, we can actually look to see to what extent do filaments contribute to the lineage statistics, and we see that they're excluded from these statistics. So the fact that we have slow-growing cells and fast-growing cells appears to not be due to going through this filamentation state. The filaments are actually quite bad, it turns out, on longer time scales in these devices. So you'll see that probably in this next case. So the last thing we've been doing...
They're not genetically different, right? Everything's used.
Not genetically different, right. Everything is identical as well.
Selection
Yeah, so selection here is
sense.
on the phenotypic diversity that's generated, right? Okay, so then we said, let's go, can we engineer a situation where we actually have states that we know, right, are just stochastically switching. So I'm not going to go into the molecular details, but we've done this now with tetracycline resistance. So we actually have a strain that switches between on and off, such that on means that you're resistant to tetracycline and off means you're not. And the switching rate between this on and off is roughly 10 to the minus 2 per generation. So that's considered a super fast stochastic switch in bacteria. The persistent switch, for example, is 10 to the minus 6. Okay, so 1 in a million switches. So this is the fastest switch we know. That's bistable. And so now you can see here free switching in the mother machine. Okay, so this cell just switched to red. This cell is green. It's making many, many divisions. And eventually you'll see, this is just free switching. You see this red cell filamented, and now it's going to die. This guy here is still green, and eventually, I believe it switches to red if we wait a little bit longer. Okay, so that's just free switching without selection at all. Okay, so you see it switched, and then eventually it's going to filament and it'll die. And so now we said, okay, let's do this in these devices where we have small populations, and let's give pulses of antibiotics. We can do this with longer timescales or short. These are quite short timescales, so every 6 minutes we give them a pulse, which just basically means that we're tuning the selective difference between red cells and green cells here. And let's just see what happens. So initially we start from growth in LB. Mainly the cells are red after some cell divisions. And now they're basically experiencing this fast fluctuating environment of 6-minute tetracycline. And you see that the cells are struggling with this. Okay, they're going into filamenting states here. Things are looking quite bad. And there's even cell death here. Now eventually a lineage here has made a switch, and it rapidly outcompetes all the neighbors. That's one sweep that we see here. And then you see this guy is just collapsing completely. This guy is now seemingly on the road to extinction as well by filamentation. This guy's also managed to produce green cells, but this one somehow, you'll see in a moment, gets rescued here by another green cell switching. And this population now also makes a switch back. Okay, so these cells are not deterministically going to win the battle, right? And if you switch back, you basically die, all right? And these filaments are actually quite bad as well because they push cells out of the way, so they take up space, but they're in fact useless. So you see that in a simple experiment, we already have, right, 2 states that we thought we were controlling, and then a 3rd state, which are these filaments, which now the other states have to fight against. Okay, so that's going to be the end. So we've been measuring these single cell dynamics of E. coli populations, and I showed that you have real responses to fluctuating sugars, of course, part of the physiology of the bacterium. Memory then becomes an important adaptation. And then if you look at lineage statistics in constant environments, you can actually predict. We've been trying to see what aspects of population dynamics can we predict, and what are the basic ingredients we need to know to predict the dynamics. So in one case, we need to know whether there's memory. Okay, otherwise it's going to change the whole behavior. In the case of if you make things as simple as possible, a constant environment, you can predict the statistics of these trees. And then if you go further and start doing fluctuating stresses, it seems that these lineage behaviors could be quite useful for inferring what's going on in the experiments. And then if you go beyond it, maybe we'll be able to measure the strength of selection in these heterogeneous populations. So I'd like to thank Guillaume Lambert, who was doing most of the experiments you saw. And the very last section was done by a student in the lab, Wei Shan, who did the stochastic switch. And also want to thank Toby Bergmiller and Kalim Jouet at ISD Austria, who provided the lack overexpression constructs for us. And then Yuichi is another collaborator that's been using this device here that showed you the constant lineages in constant environments. Thanks a lot.
Great talk, thanks. About this very last experiment, it seems like there's a genuine spatial aspect there also. So it depends very much on where the mutation, it's kind of a mutation, right? Where it occurs, whether it can be successful. It has to occur somewhere at the upper end.
Exactly.
Perhaps even non-trivial, if you know these rates, and if you know what's the advantage of the mutated one, to predict how often they switch.
Yeah, I fully agree. I mean, we're now trying to develop a framework to analyze this data and to then eventually make predictions. And I agree that space is going to take a major part of this. And if we could, of course, eliminate these filaments, we'd already make our life a little easier. But in fact, they also provide another sort of layer of, there's pushing in this device, right? There's these geometries. Do we have to model everything to predict the long-term dynamics? Hopefully not, but we'll see.
As you describe it, the phenotypic memory in the first part of your talk could be just a side effect of having proteins that last a long time. How would you go about showing that these memory phenomena are adaptations to a fluctuating environment?
Yeah, I would say as usual that's quite difficult. But in this case, these proteins, the only thing I could say to answer that would be that their degradation rates they're very, very stable proteins. So, a lot of proteins in E. coli are stable, but these are on the tail of that distribution. So again, if you increase the stability of the protein, you make it more likely for it to be useful in this kind of passive memory. It'd be interesting to look actually at if there was data on stability, right, and then looking maybe at phylogeny and stability, that could be an interesting direction.
Could you also select long-term or fluctuating versus stable response for a response?
Absolutely, I think you can do it. The only problem in these experiments is the population sizes are small, so you wouldn't be able to generate. I don't think you'd be able to generate enough mutations to really see selection. In fact, for these experiments, we can always show that the cells did not attain a mutation by simply, at the end of the experiment, fluctuating them back to glucose, okay? And then doing our fluctuating experiment again. And if they have to go through lags again, it means they haven't learned how to just grow in all environments.